Beyond the limits of our unaided vision, in seemingly mundane pond water, a whole different world exists where life thrives. A realm teeming with intricate wonders. Among these microscopic marvels is Euplotes, a ciliate that defies the simplicity often attributed to single-celled organisms. Here you can see exactly what I focused my microscope on, the edges of a tiny bubble on the slide. The light bounces off just right, giving this mirror-like effect. Even as this flatworm is out of focus in the background and starts to move off screen, you can still track it with decent focus in this reflection. But the topic today isn't about flatworms who can actually manage to retain memories after having its head chopped off. No, seriously, they're able to do that, it's insane. It's a video about these little creatures right here, Euplotes. Euplotes stands out in the microbial world due to its unique structural features, because unlike many of its ciliate counterparts that swim using mostly rows of cilia, Euplotes also has specialized bundles of cilia known as cirri. These cirri function akin to tiny legs, enabling it to walk or crawl in the water and along surfaces in its environment. Now, not all cirri are equal, and that's especially the case with these cirri near the back of the cell, known as the caudal cirri, from the Latin caudalis, meaning of the tail. They usually have between 2 to 6 of these stiff, tiny posterior spikes. The exact function is still debated, with theories ranging from stabilization in the water to possibly being a sensory structure, but on a surface level, it really does just look like pokey spikes. Even though this organism is just a single cell, it can change the way its body is shaped in response to chemicals detected from predatory organisms. By reorganizing their membrane and inner structures, they can morph their bodies to widen out, sometimes doubling their width and becoming almost circular. That resulting grape-like shape makes it harder, sometimes impossible, for predators that would have otherwise swallowed it to actually have a succulent Euplotes meal. Mine seemed to not be too stressed about the predators even with the presence of the flatworm in the sample, because these Euplotes are still quite flattened. This is the adoral zone of membranelles, shortened AZM. A membranelle referring to a collection of fused cilia that work together as a single unit to move larger amounts of water. All species of Euplotes have an AZM. The structure comprises, depending on the species, between 20 to over 70 membranelles. It extends from the left side of the cytostome along the peristome toward the anterior of the cell. These membranelles function collectively to create a beautifully coordinated beating pattern, efficiently sweeping food particles such as bacteria and algae towards the cytostome for ingestion. By the way light bounces from the movement, here you can actually see the AZM at work. This sample came from a mini ecosystem in a jar, bottled from a small freshwater pond close to my home. I'll show you that jar, a different rotifer colony jar, and my janky microscope setup which managed to get me this footage in upcoming videos. This was just scratching the surface of complexities of these ciliates, and there's tons more to know about them and their behaviors. The research papers I used for the information in this video are all in the description, as well as full text transcript of this video. Also some links to my profiles on other social media. Thank you so much for watching.